How did the characteristics of phospholipids contribute to the distinct properties of the cell membrane? Phospholipids are examples of amphipathic molecules. An amphipathic molecule is going to be a molecule that has both hydrophilic and hydrophobic characteristics. And so if we take a look at the structure of a phospholipid, what we're going to see is that we start out here with a glycerol molecule. We've got a 3-carbon sugar here. We've got 2 fatty acid tails attached to that, and it's illustrated here in this space-filled model. But fatty acid tails. Fatty acids are going to have a lot of carbon-hydrogen bonds and then carbon-carbon bonds, kind of building the structure within the length of our fatty acid. And so carbon-carbon, carbon-hydrogen are going to have about the same amount of electronegativity, and so they're going to produce nonpolar covalent bonds. These nonpolar covalent bonds are going to be contributing to hydrophobic characteristics. And so our fatty acid tails are going to be hydrophobic. They're not going to interact well with water. In contrast, instead of another fatty acid tail on here of like a triacylglyceride, we've only got 2 fatty acid tails. We've got 3 carbons within our glycerol molecule. What we've done is basically attached a phosphate group onto that last carbon. So we've got carbons holding 1 carbon holding a fatty acid tail, the 2nd carbon fatty acid tail, the 3rd carbon holding a phosphate group. That phosphate group is going to be a charged particle. Charged particles are going to be hydrophilic, and it's going to want to interact with water. And so as an amphipathic molecule, we're going to have the hydrophobic fatty acid tails, and then we're going to have that hydrophilic phosphate-containing head. And so we've got both of the characteristics within this molecule. When we put the phospholipids together to build our cell membrane, what we're going to see is that the phospholipids are going to be organized in a bilayer. And so what we've got are our fatty acid tails are going to be towards the core of the membrane. And so we've got an extracellular side outside of the cell. We've got an intracellular side inside the cytoplasmic side of the cell. But in between there, we've got those hydrophobic tails. Lots of fatty acids, lots of nonpolar covalent bonds means that we basically have the equivalent of a rough waterproof boundary. We're not going to allow for charged particles. We're not going to allow for ions. We're not going to allow for molecules that are within a hydration shell to be able to move through this, because the water that they're going to be dragging along or the charge that they have associated with them is not going to interact well within these hydrophobic tails within the core of the membrane. Now, to the external face of the membrane, to the internal face of the membrane, we've got these phosphate-containing heads. These phosphate-containing heads with their charge are going to be hydrophilic. They're going to want to interact with water. And so we're going to have our phospholipid bilayer here because of the amphipathic molecule characteristics. Our phosphate-containing heads are going to be interacting with the aqueous, watery environment outside of our cell, and our phosphate-containing heads along our cytoplasmic side of the membrane, the intracellular inside of the cell portion of the membrane, are going to be interacting in a hydrophilic environment with the water molecules along the cytosol or cytoplasmic side of the membrane. And so what we've got with this phospholipid bilayer then is establishing this boundary, which is going to block the movement of hydrophilic molecules through it. But then we've got the hydrophilic phosphate-containing heads, which is going to allow for the aqueous environment outside of the cell and inside of the cell to be able to interact with the proteins that may be kind of embedded within the membrane or other things that are found within the membrane as well.